Hello friends, welcome to BMH Learning. In this video, we are going to talk about Folch method. Folch method is used for extraction of lipids from various biological sources. The method is basically divided into two steps. In the first step, the lipids are extracted by homogenizing the tissue with 2:1 chloroform methanol mixture, and then the homogenate is filtered. In the second step, the filtrate obtained in the first step, which contains lipids as well as non-lipid substances, is washed with water. The washing frees the lipids from unnecessary stuff. Procedure. So first of all, the tissue is homogenized with the chloroform methanol mixture in 2:1 ratio. You can use 1 gram of tissue, and for that you have to use final volume of mixture 20 times the volume or weight of the tissue sample. That is 1 gram in 20 ml of solvent mixture. After the tissue is added into the mixture, the whole mixture is agitated using an orbital shaker at room temperature for 15 to 20 minutes. The agitation forms the homogenate. Now, the homogenate needs to be filtered. Either it can be simply filtered using funnel with a folded filter paper, or it can also be centrifuged for the recovery of the liquid phase. For better results, we can use both. First, the homogenate is filtered using the funnel. Now, the filtered homogenate is treated with 4 ml of distilled water to wash the solvent. Or for much efficient washing, we can also use 0.9% sodium chloride solution. Next, vortex it for 2 minutes. After vortexing, the mixture is centrifuged at low speed of 2000 rpm. The centrifugation process separates the two phases. By siphoning, the upper phase is removed. Keep this phase for the analysis of gangliosides or other small organic polar molecules. Once the siphoning of the upper phase is done, the lower chloroform phase, which contains the lipids, is evaporated under vacuum in a rotary evaporator. Or otherwise, the chloroform can also be evaporated under a nitrogen stream if the volume is under 2-3 ml. Finally, the residue left after evaporation is dissolved in 5 ml chloroform and stored under -80 degrees centigrade. While doing the experiment, keep in mind that the method of tissue disruption actually affects the concentrations of extracted lipids. So therefore, for poorly explored tissues, the analyst must be careful in comparing various methods of tissue disruption. Dichloromethane can be an alternative to chloroform and has been used in extracting lipids from samples of different natures. Use of dichloromethane can avoid the major health, security and regulatory problems associated with the use of chloroform. It has been reported that the Folch method is really efficient for the extraction of a broad range of lipid classes in human lipoproteins. This was all. Thanks for watching.